Hello students, in this tutorial I am going to show you how to do your lab number 1. Okay, so let's go right into it. So so you will need to go to this thing called the sitetoolkit.org. So once you do that, you hit enter and then you will be taken to this page. Okay, then you need to look for the actual things. Go to the left thing and then says go to the one and find the thing that says library of experiments. Okay, and then click on this. Okay, so this is like a list of your experiments that you can do. The one that we are going to be doing is down here. This one that says queuing Posner task. Okay, so click on this. And you can read the introduction yourself. Essentially, the Posner's task is basically a way to draw your attention to a very specific location, and then the attention can be drawn in location due to the appearance of the stimulus. So, and you don't really need to know like what it is. Not for your lab number 1 anyway. For your lab number 2 and lab number 3, you should know the construct much more. But then just but because of your lab number 1, I just kind of want you to get a good idea of what to write and how to write your lab report, just to get an idea. And so lab number 1 is more like a exercise, if you will. Okay, so what you need to do is go down, okay, to this thing and it says run a demo, and just follow the instruction. Okay, click to start, so you click on the screen. Okay, queuing experiment. You will see a fixation point and 2 boxes. Press on the left key, which is going to be A, if you see a go signal in the left box, and on the right key, which is going to be L button, if you see a go signal in the right box. Okay, so A if you see the signal on the left, L if you see the signal on the right. Each and every trial you will see an X, which is a Q. Do not respond to it, just ignore it. Okay, there are 100 trials. Okay, press space bar to continue. Okay, so it appear on the left. Now if you're too slow, it will tell you that you made a mistake. Okay, so you just do this for like 100 times, which is relatively quick. Okay, so I'm just actually gonna do through all of this. Okay, so at the very end, you'll see that now that you're done, did you notice that 25% of the cue were invalid? That means that they appear on the opposite side of the goal stimulus. So anyway, so you see that through this learning process, okay, you can read this yourself, okay? So basically, after you're done, you just press space to exit, and then you'll be shown this. Okay, so this is the outcome. This is your dependent variable in reaction time. So what it's basically is showing you is that in the valid trials, it cost me on average 332 milliseconds to complete the trial. But on invalid trials, which means that the cue and the stimulus, the goal stimulus is occurring on the opposite side, this is showing me 390 milliseconds, which means that there is some sort of a cognitive cost in the invalid trial when you compare it to the valid trials. Okay, so let's press spacebar to continue. And actually that is it. Okay, now if you want, you can go to the show data. It'll tell you each and every trial for 100 of them, if they were done correctly or if they're not. Now, the thing is that if there were any sort of invalid, like not invalid, but like, if there's any sort of like data that are incorrect, it's going to appear as, let me see, I think here, okay, 1, 2, 3, 4, 5. So you can see that all the ones that says 1 are correct trials over here. Okay, and if it appeared anything except 1, so for instance, 3 means that this one got timed out. Okay, so because you see this, the reaction time here was 1500 milliseconds. Okay, so you can see that the limit for each trial, the amount of time that you have to respond is 1.5 seconds. Okay, now 1 over here means that that is a correct response. You can see that all the I have a lot of correct responses, and then go down, go down, go down, go down. And 2, it would be an incorrect response. Okay. Now, there's one thing I want to add. So the way that the program shows this is calculate out your reaction time for your valid versus the invalid trial, is that they took average of all your valid trials, and then they take the average of all your invalid trials, only the correct ones, and then they actually generate the actual data. Okay, so that's how they generated the data that you saw in the previous page. Okay, that's it. So now why don't you do it and try it yourself. Okay, and when you're done, just go ahead and read the actual there's going to be a lecture on this, and I'm going to also post that on YouTube.